Hello, everybody. I am excited about today's webinar. My name is Dr. Maggie Bartlett, and I'm thrilled to be the host for the Global Virus Network Forefront of Virology webinar series. We have an exciting and informative session planned for you featuring the one and only Dr. David Markovitz. Before we begin, I'd like to thank our sponsors who made this event possible and acknowledge the attendees from around the world today and who might watch this later. We are focusing today on molecularly engineered lectins for the prevention of treatment of coronavirus and influenza virus infections. And we'll start with a presentation from Dr. Markovitz, followed by a Q&A session where you'll have the opportunity to ask questions and engage in a discussion. Just a few housekeeping items before we begin. We are recording this session, so it'll be available after the event. If you have questions during the presentation, please type them in the Q&A box. We will address as many as possible and follow up afterwards if we miss any. Please feel free to use the chat function to engage with other participants and enter your questions. Dr. David Markovitz is a professor of internal medicine in the Division of Infectious Diseases at the University of Michigan, with additional roles in cellular and molecular biology, cancer biology, and immunology. After earning his MD from the University of Minnesota and completing the training at the University of Rochester and in Lineberger Cancer Center, he joined the University of Michigan, where he has advanced to a full professor and has explored virus-host cell interactions and the mechanisms of viral oncogenesis, the role of endogenous retroviruses in human diseases, and an entire decorated career on antiviral therapies. And without further ado, I'm very excited and honored to have you with us today. The floor is yours.
Thank you very much, Maggie. And I want to say it's really a pleasure and a treat and an honor to have a chance to address the GVN, terrific organization. And I've had some wonderful interactions over the years with GVN, and I'm very happy to have the opportunity to give this talk. I'm also happy that my electricity didn't go off as it did several blocks away from here. So so far, so good. So we'll hope the weather continues to cooperate. If you hear me utter a very loud swear word, I'm sorry. That would mean that my electricity is just about to go out. So hopefully I won't be doing that. As Meg said, I'm going to talk today about some very fun things we've been doing now for a pretty long time with lectin. By the way, and for those of you who are retrovirologists, you probably think that lectins are simply exist in the world to activate T cells so that you can study how HIV and other such viruses replicate. And basically that's how I was in about 2007 or 6 or something like that. So my interest in lectins for their own sake as an antiviral agent came after a long time with doing other things. So the bottom line is lectins are proteins that are often, we have them in our own human bodies, but the ones we'll be focusing on and that people focus on most in therapeutics come from plants, such as ours that come, or fruits, such as ours that comes from banana, found in plants and animals. And the key thing about them is that they have evolved over many millions of years to recognize very specific types of glycans. So they're highly specific, and many pathogenic viruses have glycans on their surfaces, on their surface proteins, that are not found on our normal healthy cells. They are found on some cancers, by the way, which is also a target of our work, but I won't be talking about that today. So the idea is that if you can use a lectin to bind to one of these viruses that has an abnormal glycan on its surface, then the virus will no longer be able to enter into a human cell and won't replicate. This idea was not originally ours, of course. People have thought about this for quite a long time. I'll show you some stuff that is original to us, though. So Banlec, which is the protein we're going to talk about, is a lectin found in the fruit of ripened bananas. And in fact, our very first paper on this topic, which I won't have much time to get into today, was done with banana lectin, Banlec. It was actually purified directly from bananas by our collaborator then, the great glycobiologist, Erwin Goldstein. And he was already pretty elderly, and he and his equally elderly colleague would go to a Kroger grocery store, buy the bananas, mush them up, and extract the banana lectin. The work I'm going to show you today, because I'm the next generation down from Erwin, is all done molecularly. So anything I show you with lectins are lectins that are cloned, Banlec that's been cloned into E. coli and produced that way. So Banlec is a member of the Jacqueline-related lectin family. It's a monomers 15.6 kD approximately. I'll show you later. It's usually a dimer, but occasionally a tetramer. And it can bind to high mannose structures that are on several important pathogenic viruses, but high mannose is not found hardly at all on normal, healthy human cells. So that's how it discriminates viruses from us. And again, I just want to emphasize, high mannose is not just any old mannose. It's not one mannose or two mannoses. It's 5 to 9 mannoses on a residue. So we published a paper showing that we could actually treat HIV with Banlec. That was very nice. It was a nice paper. We showed that it worked the way we thought it would. It actually, for better and for worse, drew a lot of attention from the world press because we were talking about using it as a vaginal microbicide. And the combination of sex, bananas, and HIV created quite a commotion in the world press. And we were quoted and misquoted in many places. And I got called from famous HIV-infected people asking me if they should eat bananas. I said, don't bother, you won't absorb the banana lectin. Anyway, so however, all this was very interesting. It taught me always to do a press release when you have anything interesting, because that way you won't be misquoted. And then the second thing was, though, that in spite of the fact this was really interesting and fun, the way the banana lectin comes out of a banana, it can't really be used therapeutically because it's mitogenic. That's why we use it, right, to activate T cells. But that is harmful in an animal and a patient because it can, it indiscriminately activates the immune response. And so we thought that we could not give regular Banlec to people. We subsequently have data actually showing in mice that that's actually true, that the wild type banana lectin is indeed harmful. So as good luck would have it, this was God speaking directly to my very brave graduate student, Mike Swanson, and suggesting to him that he could actually molecularly modify Banlec to remove mitogenicity and retain antiviral activity. And no one had ever done this up till this point. So it did take some daring or naivete on our part. I'm not sure which it was. But essentially, through such engineering, Mike was able to show the first demonstration that two functions of a lectin can actually be separated through targeted engineering. This was made possible partly by the fact that we knew the structure of Banlec as determined by Jeannie Stuckey here at the University of Michigan. So it turns out that our Banlec that we engineered, where we took the histidine in position 84 and changed it to a threonine. And by the way, it matters not only that we got rid of the histidine, but what we changed it to. And I don't really have time to go into all the structural biology, but you can read about that if you are quite interested. But anyway, it turns out that our H84T Banlec retained broad spectrum antiviral activity. It was highly active against all strains of influenza tested, at least to that point. I'll come to a little bit of a ripple in that discussion later. All HIVs we tested and all hepatitis C viruses that we tested. And it also, we could show in vivo, it blocked vaginal transmission of HIV. And if you put it in the nose of a mouse, it could block and even treat infection with H1N1, and later we showed H3N2 influenza viruses. So many of you who are researchers know this feeling that the guy with the knife in his hand is actually the editor of a major medical journal, and the person being held down is not actually Isaac, but instead either you or one of your postdocs or one of your students. And that's how the review process can feel, but fortunately in this case, we had good results, and we published this in Cell with 26 authors from 5 different countries, because we not only showed all this activity, these activities, and looked at it from a glycobiology point of view, but also looked at this from 3 different structural modalities, figure out why the mitogenicity had gone away. And I just don't have time to get into that here, but say if you're feeling very brave, read the original paper, and you can see that. Now, subsequently, in some further work we did, and by the way, I just can't emphasize enough that all this work is done with many people, and I'm going to try to highlight some of them, and I'll mention them at the end. But this has been a very big project with lots of collaborators and enthusiastic and wonderful collaborators, and from multiple different countries. So we then had a paper where we showed that H84T Banlac was, again, highly effective against influenza. This was done by Ivi Kove-Datsun, who is a MD PhD student in my lab, and is actually now back at Michigan as an infectious diseases fellow, to my happiness. What Ivi was able to show, and working with others, was that H84T not only could be given in the nose, but could actually be given systemically, safely, and to treat influenza infections. And this could be done as long as even 72 hours after the initial viral challenge. She was also able to show that you can give it subcutaneously, although it wasn't quite as effective. And one of the more important figures here is figure C. And the reason that's important is that many people ask us, well, if you give this protein to an animal or a patient, won't you develop antibodies, and won't that harm the animal, and won't that destroy the molecule? So the answer is shown here, which is we gave H84T to mice, we let them develop antibodies, then we challenged them a month later and treated them, and the treatment was still totally efficacious, and there were no side effects from the banana lectin. Now, the mice definitely do develop antibodies, but probably they're just not harmful. And in fact, people develop antibodies to Banlac from eating bananas, but it's IgG4, so probably tolerogenic in most cases, not all cases. So why is H84T so specific? Again, it recognizes the viruses, but not healthy human cells or animal cells. This finding was consistent with the literature. We've also given H84T Banlac to all sorts of different mice, immunocompetent, SCID, reconstituted with human immune system, hamsters and rats, and it's always been well tolerated. Significantly, we also, through the contract NIAID, graciously paid for us to have a company check. And what they did is they took 35 different tissues from 3 different individuals, and in each case of 3 different species, humans, monkeys, and rats, to see whether H84T recognized normal healthy tissue. It did not. The only place it did have some recognition was of a few hematopoietic cells. We'd never seen problems with hematopoiesis in mice given H84T, but we decided we will give it to rats just to see, because those had been the object of these staining experiments. The rats did not develop any problems with hematopoiesis. If you're one of the conoscenti, they did develop a little bit of an elevated MCV, so their red cells were a little big, but there was no anemia, and there was no increase in an unhealthy way, polycythemia of red cells. So we were busily working away on influenza, At that point, the next plague came along, and that naturally was SARS-CoV-2. As good luck would have it, we'd already been interested in coronaviruses, working with Jasper Chan and KYUN in Hong Kong. And the reason being that they have high mannose on the spike protein. So we thought, maybe these will work against coronaviruses. And in fact, it does. So sorry, these next three figures have lots of data, but I'll try to just summarize it. Essentially, it turned out that H84T was highly effective, first of all, in vitro against all coronaviruses tested. So that included endemic coronaviruses of the type that don't usually cause disease except if you're immunocompromised. It included MERS, the very deadly Middle Eastern version of this, which is a 35% mortality rate. And then against the original SARS, it was effective. And by the way, when I say effective, I'm talking about in the low nanomolar range. So it's really quite effective. And then when SARS-CoV-2 came along, we tested it, and it too was very sensitive to H84T band-like in tissue culture experiments. And that's measured both genetically, so copies of virus, as well as plaque reduction assays. Probably yet more significantly, using systemically administered H84T, we were able to show that it was highly effective against MERS in vivo. So there are a lot of figures here, but we did some ex vivo studies, but probably the key thing to look at is C here, because there we are taking mice and giving them MERS. By the way, that's a whole big deal because it turns out, the receptor for MERS is not ACE2, but rather CD26. And human and mouse CD26 is not susceptible to MERS infection. So these are knock-in mice. It took about 2 years to generate with human CD26 in order to do the experiments. And so what you can see in C is if you don't treat these mice, they all die. But if you treat them with H84T, about 80% of them live. And the corresponding and expected pathology and virology changes in the tissue are shown below. And those fit very well with that, as well as viral loads also corresponding. So this was very, very effective. Finally, when it came to SARS-CoV-2, we could actually give H84T, and here we switched to a hamster model. So this isn't a lethal model. We haven't ever done this with the ACE2 expressing mice that you need for a lethal model, but this one probably, the hamster model probably resembles human disease more readily. But both models, of course, are good for different things. But what we were able to show is whether you gave H84T intranasally or you gave it systemically, and whether you gave it before challenge or about 24 hours, started about 24 hours after challenge, you could very effectively treat SARS-CoV-2 when measured by pathology changes, viral load as measured, through PCR, and also, again, using plaque reduction assays. So we were very happy about this, naturally. And we also were wondering about other variants, because as time went along, of course, the SARS-CoV-2 evolved. We haven't, I think, tested it against the very most recent versions, but I think you'll see in a minute that it's highly likely to work against those. But when we tested Delta and Omicron, we were able to show that, again, these viruses were sensitive in the very low nanomolar range of H84T. So again, this was very, very happy event. So I won't go through all of the details here, but this was some beautiful work done by Peter Hinterdorfer in Austria and his collaborators. And essentially, we wanted to look, we'd really found when we did our first paper on H84T, the strength of doing structural and physical biology and biophysics is wonderful because you can really explain what's going on. And that's extraordinarily helpful on every level, not to mention just fun and interesting, which, you never want to overlook things that are just fun and interesting. And this was really cool, and it explained a lot to us. So we did these studies looking at atomic force microscopy and other methodologies that I'll mention in a second. And we're able to very nicely visualize exactly how H84T interacted with the trimer, the spike trimer of SARS-CoV-2. I should point out that one of the interesting findings for the conoscenti again was we had originally said that H84T was a dimer. Another very strong group came out and said, no, it's a tetramer. And so with atomic force microscopy, we could clearly see what the answer was, which was that 50% of the time it's a dimer, a fourth of the time it's a monomer, and a fourth of the time it's a tetramer. So everybody was at least a little bit right. The modeling suggests that it works better as a dimer, though, and I think that that's probably the dominant thing, and that's what we're using going forward with our work. As I say, I don't want to go into everything here, but using plasma resonance, atomic force microscopy, and single molecule and dynamic force spectroscopy, all very useful things. We found that the bonds between H84T band length and the trimeric spike protein of SARS-CoV-2 were strong and diverse. And some of them even extended from one of the monomers to the other monomer. So it was really very cool. Again, if you're very interested in this topic, you'll be surprised to find out that the interaction was not with the receptor binding domain. So it was not with the part of SARS-CoV-2 that spike that interacts with ACE2, the receptor for it. But in fact, it's with other parts of the molecule. So that was surprising. In fact, when we first were wondering if we were doing some experiments right, and it turned out we were, because this was the answer. The other thing is that H84T not only has very strong bonds that are chemically strong, but they're in diverse sites of high manos. So there's at least 5 high manos sites that are bound by H84T. One with 9 manoses and the other 4 with 5 manoses. This likely accounts for the broad spectrum of activity, but also probably why when we tried to generate resistant virus, we couldn't do it. Now, we haven't extensively and endlessly tried that, but at least at first blush, it's very hard to generate resistant SARS-CoV-2 or other coronaviruses when it comes to H84T band length. So the crucial thing at this point, we had a paper that Jasper Chan was the first author in Cell Reports Medicine. This broke my previous record. We had 40 authors from 4 different countries and 3 senior authors, of which I was the senior of the senior authors. Amusingly enough, KYUN, who was one of the senior authors, was the guy who discovered the original SARS. And Josef Penninger, another one of the authors and an important collaborator of ours, was the person who originally discovered ACE2. So we had some really interesting synergy there, coincidentally. So what we were able to show is that systemic and especially intranasal H84T band length, which has broad spectrum anti-coronavirus and anti-influenza virus activity in vitro and in vivo, could be useful for the current epidemic, as well as being ready for future pandemics that might be due to SARS-CoV-2 or other coronaviruses that emerge and emerging influenza viruses. Although there's a little wrinkle I'm going to show you in a minute having to do with the influenza viruses. So up until this point, I don't want to say that H84T always worked against everything because it didn't. For example, some important viruses that we thought would be susceptible, such as respiratory syncytial virus uh, and hepatitis B virus, were not susceptible to H84T. And that was a surprise because they have high mannose on them, but we just that's what the facts showed. There's also some gray zone because, for example, originally when we did herpes simplex virus, also expected to be susceptible, it was not. But then when we worked with a really good group in Syracuse, and they headed by Jennifer Moffat, uh, I don't know if it was the skill of the group or the fact that they used different cells, but there we showed very clear activity against herpes simplex, CMV, and also VZV, and even in some animal models with human skin models. So, but up till this point, very recently, H84T had worked against all influenza isolates we'd ever tested, including H5N1, which of course has now become an incredibly important issue in the world. And that original experiment that we published was done with pseudotype virus, so not with wild type virus. So much to my unhappiness, when we tried H84T, this was done by Jasper Chan and his group, against the new highly pathogenic H5N1, it was resistant to H84T. So I was surprised and not in a happy way, but those are what the data showed. However, as good luck would have it, long ago when we first produced H84T, we produced 9 other mutants of histidine 84. The reason we did that originally was to understand using NMR the structure and function of H84T. This was done with Hashim al-Hashimi, when he was back in the day when he was at Michigan. So we did that, and there was one other mutant, H84G, that was highly active against, at least against HIV, and had lost mitogenicity. It was the only one of those 10 mutants other than H84T that had that profile. And for years, I would write in grants, well, if we need to, we can look at H84G. And one day I said, really, it's time to look at H84G, which turns out to actually be more active against certain cancers, by the way. And it's 3 to 5 times more effective against SARS-CoV-2. But really, we didn't need much more activity against SARS-CoV-2, but we did need more against the new H5N1. And sure enough, it's quite active. IC50 of 132 nanomolar, which is similar to what we'd seen before with H1N1, where we know it works in vivo. So H84G and H84T are presently being tested for in vivo efficacy by Jasper's group, and we're hopeful that it'll work. We expect that it'll work, and then we'd be able to hopefully be part of a large-scale assault on the new virus. So, but stay tuned. Now, again, this whole topic of lectins was totally new to me about 2008. But having done this really cool molecular engineering of lectins has gotten somewhat contagious. It's addicting because it's really exciting to do. There's very few groups doing it. Barry O'Keefe at the NCI is probably the other leading person doing this. And who's the guy who's done all the work with Griffithson. Or a lot of the work with Griffithson, and which is another high mannose binding lectin that looks promising to be used therapeutically. We thought maybe we can make H84T and H84G even better than they are. And that's a little bit of hubris because when you think about these proteins having evolved over millions of years in plants just to bind high mannose, you figure, well, gee, can you really improve it? So, but we're trying. This is done with Wenjing Wang, who's at the University of Michigan. And Wenjing has this wonderful way of system whereby you introduce a series of random mutations into the molecule of interest, in this case, H84T or G. And then you express it on a yeast library. It turns out, by the way, it looks like to express it, you really need to force dimerization, interestingly enough. Otherwise, it doesn't express well. This is a theme that's coming up in some other areas we're working on too, where you really need forced dimerization to make certain things work with this lectin. But then she's going to screen actually a whole H1N1 that's of course been inactivated to see whether we can, any of these variant mutants can now bind better to H1N1. And the idea would be most likely, if it does, it's due to differences, that it's binding to the hemagglutinin, which is, I think I neglected to mention before, Evie Covey-Dotson had shown in the old PNAS paper that the way that influenza is inhibited by H84T is that H84T binds to hemagglutinin, HA, and then that inhibits fusion of the membrane. More than attachment, it inhibits fusion. Some viruses are more inhibited at the fusion step, some are more inhibited at the attachment step, if you're especially interested in these types of questions. But by using the whole virus, we'll also perhaps figure out if other proteins like neuraminidase, for example, has high mannosonic too. So it's a potential target, although not as attractive as HA that's on the surface of the virus. All this by way of saying we hope we're going to emerge with yet improved H84T and H84G using this directed evolution. So with apologies to the gods, we hope that will actually emerge. So we, of course, really want to move this to the clinic. I have a wonderful collaborator at Michigan, Ashley Brownell, who's in the pharmacy school. And she's working very diligently with a student to make an intranasal formulation that can be used really well in the clinic. She's working to make it something that is very stable, that could be disseminated widely in the world, that won't need a cold chain, that won't need sophisticated healthcare to deliver, so that people everywhere in the world would be able to put this intranasal formulation in, and that it would work for at least a day, so you just have to give it once a day. That's probably going to work because even just putting it in H84T in with in PBS, it actually stays in the nose a pretty long time. It turns out, unbeknownst to me, there's a whole science behind all this. There's lots of complicated chemistry that you really need a collaborator. Let me say that. If you're ever trying to do this, you really need a collaborator if you're a mere virologist. It's a very interesting process. Of course, then we would need to do formal toxicity studies. We have lots of informal tox, but you have to do formal tox if you want to move ahead. And you have to be able to make this GLP, GMP production. So this is, sort of the classic valley of death. Somebody has to pony up a couple $1 million before you can go into clinical trials. The good news is if we can get that far, then the first studies in people can actually be done with influenza challenge. As you know, people do these studies, and we have collaborators both at NIH and in Hong Kong who could do these studies where we would then actually challenge healthy young volunteers with flu and see whether this works. So it's great, and we could also go on to do studies with coronaviruses. At some point, we want to examine aerosol delivery also as a way to treat. And our data so far show that you can probably give this safely to people systemically who are really ill. So we'll have to see if that'll be a higher bar, of course, a higher threshold. We might be able to also get there through the fact that we're trying to develop some of these same approaches for treating various forms of cancer. In fact, my collaborators really made a CAR T cell that is steered by H84T rather than by an antibody, and it kills pancreatic cancer in vitro and in vivo. And we published that. That's with a group at Baylor, Katie McKenna. Anyway, so that's where we're going with that. So just to stop for a moment, I've mentioned a number of the people involved. Evie did a lot of the influenza work. Dan Boudreau, who's now an Army scientist, was the guy who made all these different mutants of banana lectin to try to do these structural studies and ultimately come up with H84G. Maureen Legender is my long serving and suffering research associate and lab manager who makes a lot of the lectin so it can go to other people. I mentioned Ashley and Wenjing. I mentioned Irwin and Jeannie and her colleague Jennifer, who have done the structural biology. I didn't have time to talk about some beautiful work done in the lab of Anna Smed Sorensen by Meng Yu and with my longtime friend Karen Laurie, which came to a really exciting finding, which is that if you treat human, this is done with human cells, with dendritic cells presenting and T cells involved. And what happens is and B cells, and what happens is if you treat influenza infection in that setting, now with H84T, it kills most of the flu, but a little bit of it is left. And that little bit is enough to stimulate the immune response. So then it appears that perhaps part of the effect of H84T is actually allowing for a favorable immune response to influenza, something I used to poo-poo all the time. I'd get grant reviewers who'd say, what about the immune response? I'll say, that's all wrong. But turns out those grant reviewers did know something. And it's a very nice. The other really exciting thing is it looks like it also helps to preserve the secondary immune response. So, for example, when you then challenge with cytomegalovirus, the immune response is much stronger. So the hope is that this will hold up, say, for pneumococcal pneumonia, because, of course, a fair number of people who die of or get very sick with influenza infections, it's because they get a secondary infection with a bacteria like pneumococcus. So if this sort of treatment also preserves the response to pneumococcus, that will be lovely from a clinical point of view. Charlie Rice did the hepatitis C work I talked about. This is not why he won a Nobel Prize, I'm sorry to say, but he did indeed win, and he's a wonderful guy. So once in a while, the good actually prosper. It's great. Bart Tarbert did a lot of the work with flu, and Elke Lipke was an important collaborator here at a company in Ann Arbor. Jasper Chan, I mentioned KYUN. Very younger colleague Ken, who's been doing the work very recently with the coronaviruses and with flu, very nice. I mentioned Peter Hinterdorfer before. Hashem was vitally important. He's a brilliant really biochemist, structural biologist who we had the good fortune to have at Michigan before. And when you're a virologist trying to do structural biology, it really helps to have a very terrific structural biologist as a friend and collaborator. Scott Evans at MD Anderson was important in some of the work with intranasal work. Dominique Scholes has been a longtime friend and collaborator who's done some of the work looking at mitogenicity and lack thereof. Hans-Joachim Gabius was a fantastic glycobiologist, an extremely generous collaborator who unfortunately passed away at an all too young age, but he was just vitally important in this work. I mentioned Joseph before, who has been very important in the glycobiology, and Johannes Studelman is his younger colleague who's been vital also. My longtime wonderful friend Ramesh Akina actually did some of the mouse studies, and we've had many, many other collaborators who I can't possibly mention on one slide or in one talk. And of course, all this requires funding. We got MTRAC as a state of Michigan program to help get things into the clinic. DITRA is, of course, the Army. They funded some work on Ebola virus that I didn't get to show you. Basically, H84T works against Ebola. but not nearly as well as it works against some of these other viruses. The Forbes and Frankel money was actually for cancer and H84T, but there's so much overlap that it's worth mentioning them. And the NIAID, both in terms of some work that was just paid for by them and then through grant funding also. So I haven't had any opportunity to notice what time it is, but I hope it's not too late. And I thank everybody for their attention. I'm happy to try to answer your questions. Thank you.
No, thank you so much, David. You are perfectly on time, just
Great.
enough questions. Yes.
Years of giving talks, inner
Yes.
clock.
Yeah, you can sense it. I'll invite anyone listening to type questions in, and I'll go ahead and get us started. You outlined what was a really thrilling story about how Banlec became an option and that it's pretty close, it seems like, to clinical applications. Would you detail how far from being an option it is on the horizon?
Yeah. So, so much of this depends on raising money.
Yeah.
But in my undoubtedly biased view, really, we have enough data with H84T, and I think their H84G is likely to be so similar, we could go ahead with that directly also. And really, all we would need to do, I think, is make enough of it with GMP methods and do formal toxicity. So if somebody wrote me a check for whatever that would cost, $1 million, $2 million, tomorrow, we could probably go into a clinical trial in 9 months, 6 months, 9 months, something like that, 6 months.
Wow.
And we have people who would do it, but getting somebody to write that check is not easy. We are talking to people in the government. I'm sure it comes as no surprise to virologists, but it's hard to get investors into virology because it's harder to make a profit, harder. And beyond that, it's way harder to do the studies, but I think here, because we could do a challenge study, and we are talking about doing it in the nose, I think it'll be easier. So it just depends really when we can raise the money, but we're very dedicated to trying to do this. I had a company for a while that didn't end up prospering. Probably the most interesting part of it. It did help, and it was very fun experience. It was right in the middle of the epidemic, so rather than just sitting around at home being depressed, I could work on my company. I was also seeing patients who had COVID, so in spite of the fact there's a certain small amount of risk, it was way better than sitting around at home being depressed. And but the amusing thing is the guy who put up the money was Sam Altman, god of AI. So anyway, so that was kind of an interesting side excursion. So anyway, that's what depends whether we get the money basically, and I believe if we get the money, it'll have to probably be from a government source.
Sure, and with everything going on with H5N1 and H5 circulating in a lot of mammalian species in the U.S. and Mpox being declared a public health emergency of international concern. I don't know if you have any data on if this would be useful in that case or not.
Yeah, so Mpox supposedly has high mannose on its surface.
Right.
And we're trying to see if we can test this against Mpox, but I have to find somebody who's got Mpox and is willing to do it. If
I've
somebody
got a
out there,
for you. Yeah.
Great, because we are happy to send this to somebody who, if we have somebody who's doing Mpox and they have it growing in tissue culture, it's really easy to send them. We would send them both H84T and H84G probably and just see which one worked. The good news about them at this level of work is they are easily made in bacteria. So we have no trouble having active collaborations with people. So if there's somebody out there, or if you can introduce me to somebody, that would be great. We'd be happy to.
I will follow up after this. I'll email introduce you, but we're actively moving to help on that front and pandemic influenzas right now, as you can imagine.
Great. Yeah, we're very open to any suggestions or funders or anything like that, for doing that. So, but we have had some interest anyway from parts of the government. So I hope it'll go beyond interest and into funding. So.
No, sure. And like you said, it's got two potentials. I thought it was really intriguing, the story about how it was originally isolated. And I wonder if there are other interesting lectins that could be isolated that maybe we just didn't have someone bought a banana. And
that's very true. I mean, and of course there are, Josef Penninger's group has been making lectibodies. These are where you have the FC of an antibody, and then you have, instead of the two FABs, you have the lectin molecule. And they made a complete library of every mouse lectin, and now they're working on every human lectin to see whether in mice and human there are therapeutically useful lectins that haven't been discovered. Plants, I mean, there's this whole area of actually Barry O'Keefe came at this from a natural products point of view, which natural products are very cool. We have a guy here at Michigan, David Sherman, who has made a brilliant scientific career by going diving in all sorts of exotic locales to bring up new things from the sea bottom and test them for antibacterial activities. So it's a
pretty fascinating.
Yeah, it's a pretty fun way to make a career, and his science has been outstanding. So anyway, so yes, there's probably all these things are hiding, and finding them is the question.
No, but also to see something that has such potent antiviral activity in a number of really important pathogens that we're seeing trouble keeping up with vaccination rates. And so having some of these other, you said in mice, up to 72 hours, it was effective after infection.
The only caveat with that is that, what do you call it? I'm blanking out on the Tamiflu was also effective at 72 hours. And clinically, it's not clear that Tamiflu is actually effective at 72 hours. So, you have to take that part, I think, with a small grain of salt, but certainly it was better that it was effective than it wasn't. So.
No, thank you so much for this engaging conversation and webinar. I've learned a lot. I'm sure all of our listeners today and in future days will gain much knowledge on VanLek and the work that you've done in this space.
Thank you. Thank you.
Oh, and I think somebody might be trying to type me a question, so I might give it a minute, but we do hope everybody joins us next month for our next webinar. And of course, the GVN annual meeting this year is in Durban, September 16th, 18th. And I'm very excited that we'll be there, and hope everyone here can join us. Oh, I do see, sorry, now I'm getting them. I am so sorry for the two questions. Sten Vermond, the GVN president, asks, do you have any structural biology speculation as to why mutant VanLek did not affect hepatitis B despite the abundant mannose?
Yes, so a very good question. I don't have a structural explanation for this, but our recent experience with H84G has led me to want to revisit the whole idea that we might be able to treat hepatitis B with it. There's also the issue of, again, if you don't use exactly the right cells. Well, the hepatitis B was done by an outstanding laboratory, so it's probably true it doesn't work. But we haven't looked at how it fails to interact with protein. That's tough to do. So I don't know for sure. I am very interested in why H84G is working against H5 and H84P isn't. So I think we're trying to re-engage our collaborators on atomic force microscopy to look at that, because there you have two extremely similar molecules that exhibit a pretty significant biological difference. So,
No, absolutely. And then Dr. Ralph Walker is asking, do you know of Atenu Biotics Incorporated, who has a novel broad spectrum antiviral?
I don't know of them, but apparently I should.
Yeah, yeah. Well, thank you for bringing it to our attention.
Yeah,
Um,
maybe you can email me something about them. What
yes,
do we know what the agent is like? I mean, what is it? Maybe.
I'm not sure, Ralph, if you can type more information. Otherwise, I'll look this up and share the link with you, David. And
Yeah, that'd be great.
sure. Um,
Yeah, I
Ruby,
mean,
can you send our email
obviously,
to Ralph?
I mean, broad spectrum antibiotics obviously are a bit of a holy grail, and it's very hard to make them in the typical way because of the enzymes that replicate these viruses have differences, but a lot of, some similarities, but a lot of differences. So I think it's been very hard to actually produce broad spectrum drugs. That's why attacking shared surface markers is potentially of interest. Now, to me, the holy grail of this would actually be to design a small molecule that recognizes high
Oh,
mannose.
yes.
And I'd love to engage that. So you probably need AI plus. But you couldn't have done it even with AI a couple of years ago because I think there was no agent. But now one group did find a small molecule that does seem to engage high mannose. So now you can imagine riffing off of that and making it better. So
No, completely. I think the way in which technology has exponentially increased gives us a lot
yeah,
of cool new things to chase down. So
yeah,
certainly following up on small molecules to mannose would be a cool avenue to chase.
yeah, yeah. I find that extremely interesting. So
I think that was all our questions, but I really enjoyed our conversation today, David, and
thanks,
I
Maggie.
look forward to sharing and continuing to connect and maybe getting to see what Van Leck does to MPOX.
Sounds good. Thank you very much.
All
I
right.
appreciate it. And bye bye.
Thanks, David. Thanks, everyone. See you next time.